How did your prehistoric ancestors know whether a water source was a life-giving spring or a deadly poison pit long before the invention of microscopes, chemical test kits, or modern sanitation science? How did early humans survive for hundreds of thousands of years in environments where drinking from the wrong puddle meant an agonizing death within 48 hours? To solve this evolutionary survival mystery, we must first dismantle a dangerous modern misconception. We often assume that clear water means clean water, and that dirty water is obvious to the eye. In the wild, this assumption is dead wrong. Some of the clearest, most inviting pools in prehistory were concentrated pots of deadly cyanobacteria, toxic heavy minerals, or invisible parasites like dysentery and liver flukes. Conversely, muddy river water stirred up by a passing herd of zebras might be completely safe to drink once settled. Ancient humans could not afford to guess. In the Pleistocene wilderness, a single mistake meant severe dehydration from diarrhea, organ failure from heavy metal toxicity, or lethal parasitic infection. To survive, your ancestors turned into master forensic investigators, reading the landscape like a crime scene. They developed a complex system of environmental tracking that combined animal behavior, plant indicators, geology, and microsensation to test every water source before a single drop touched their lips. The foundational scientific proof for this ancient water wisdom comes from the extensive archaeological research of Dr. Richard Potts, director of the Human Origins Program at the Smithsonian Institution. Dr. Potts spent decades excavating the famous Olorji Sali Basin in southern Kenya, a region that experienced extreme climate fluctuations over the past million years. His empirical findings revealed that early human species, like Homo erectus and early Homo sapiens, did not settle randomly near any waterhole they found. Instead, archaeological sites show clear evidence of selective land use. Ancient humans repeatedly bypassed large, permanent lakes in favor of small, ephemeral upwelling springs located miles away. Why would thirsty hominins walk past a massive lake to fetch water from a tiny trickle in the rocks? Geochemical analysis of the ancient lakebeds revealed the answer. The large lakes were highly alkaline, salty, and packed with toxic volcanic minerals, while the small upwelling springs provided pure, filtered groundwater. Early humans were actively evaluating water quality across vast landscapes and making deliberate choices based on environmental forensic evidence. How did ancient humans execute this forensic water analysis without modern laboratory tools? Their first line of evidence was animal tracking. Animals are walking biological sensors. Different species have vastly different tolerances for toxins, salinity, and parasites. When an ancient tracker approached an unfamiliar waterhole, they did not look at the water first. They looked at the mud surrounding the banks. They examined the diversity of animal tracks pressed into the earth. If a waterhole showed prints from a wide variety of species, including sensitive herbivores like bushbucks, baboons, and fruit-eating birds, the water was almost certainly safe. Baboons in particular share a highly sensitive digestive system with humans. If a troop of baboons was drinking comfortably at a pool, early humans knew the water was free from lethal toxins. However, if the tracks around a waterhole belonged exclusively to hardy scavengers like hyenas, jackals, or vultures, alarms immediately went off in the tracker mind. Hyenas possess stomach acid so strong that it can digest rotting flesh and neutralize heavy bacterial loads that would kill a human within hours. A waterhole used only by hyenas was a clear signal of contamination. Even more crucial was the physical condition of the tracks themselves. Master trackers did not just look at who visited the water, they analyzed how the animals behaved after drinking. In his pioneering research on traditional tracking techniques among the San people of the Kalahari Desert, anthropologist Dr. Louis Liebenberg demonstrated that human tracking is an advanced form of scientific hypothesis testing. A skilled tracker can read the subtle story written in the dirt. If the tracks leading away from a pool showed a smooth, energetic stride, the animal was healthy. But if the prints showed an animal staggering, dragging its feet, or collapsing nearby, the tracker knew the water contained acute nerve toxins or deadly blue-green algae. The perimeter of a toxic waterhole was literally a forensic crime scene, littered with the tracks and bones of animals that drank and died. To understand how these forensic clues were applied in real time, let us reconstruct a concrete scene from 40,000 years ago during a brutal dry season in the East African savanna. Imagine you are a 20-year-old hunter walking alongside your uncle, an elder tracker whose face is weathered by decades of sun and wind. You have been tracking a herd of eland for 2 days, and your water skin made from cured antelope's stomach is completely empty. Your throat burns, and your muscles ache with fatigue. As you crest a stony hill, you smell the faint, damp scent of moisture. Below you lies a secluded basin filled with a dark pool of standing water shaded by a grove of fever trees. You sprint down the hill, desperate to plunge your face into the cool water, but your uncle grabs your shoulder with a grip like iron. He points his wooden spear at the muddy bank. He does not say a word. He forces you to look at the ground. You look closely at the wet mud. You see no bird tracks. You see no baboon prints. You see only the heavy, dragging tracks of a single old zebra that walked into the water 3 days ago. Your uncle walks slowly around the perimeter of the pool, pointing his spear at small details in the dirt. He points to a spot where the zebra tracks suddenly become erratic, twisting sideways as if the animal suffered from violent muscle spasms. 10 yards away, half hidden in the tall, dry grass, lies the carcass of the zebra, its belly bloated and untouched by predators. Your uncle kneels beside the water, but he does not drink. He scoops up a handful of mud from the edge and brings it to his nose. He smells it carefully, detecting the sharp, bitter stench of sulfur and decaying organic toxins produced by microcystis algae. He looks at the surface of the water, pointing to a faint, iridescent green film floating along the shady bank. He reaches down and picks up a small gray river stone from the water edge and flips it over. The underside of the stone is clean, completely bare of caddisfly larvae, freshwater snails, or small water bugs. The absence of aquatic life is the ultimate biological proof. The water is a silent killer. Your uncle tosses the stone back into the pool, stands up, and gestures toward the distant mountains. He knows that 6 miles away, near the base of a granite cliff, grows a thick stand of wild fig trees. Wild fig trees require deep, flowing freshwater springs to survive. He chooses to walk 6 more miles in agony rather than drink 1 mouthful of the sparkling pool before you. That forensic decision saves your life. This leads us to the second layer of ancient water forensics: botanical and geological indicators. Plants are stationary biological monitors that spend their entire lives filtering the soil and water beneath them. Early humans possessed an encyclopedic knowledge of plant ethnobotany, using specific vegetation as visual maps to locate clean underground aquifers. Groundbreaking research conducted by Dr. Amanda Henry, an expert in prehistoric diets and plant microfossils at Leiden University, has shown that ancient humans possessed an extraordinarily sophisticated understanding of plant ecology. By analyzing microscopic plant silica structures, known as phytoliths and starch grains, preserved in fossilized human dental plaque, Dr. Henry proved that early humans systematically harvested plants that grew exclusively near clean, flowing water sources. Plants like wild celery, watercress, cattails, and specific willow species were not just food sources, they were living proof of safe water. Willows and poplars contain natural salicin, a chemical compound that acts as an antibacterial agent, and their roots act as natural biofilters, purifying the surrounding groundwater. Conversely, ancient humans learned to avoid water holes surrounded by certain salt-tolerant or toxic plants. In arid regions, water pools surrounded by salt bushes or stunted pickleweed were known to be dangerously saline or packed with magnesium salts that acted as severe laxatives, causing rapid, fatal dehydration. Furthermore, early humans understood geological filtration. They knew that water oozing directly out of clean sandstone or granite cliffs was pure, filtered by hundreds of feet of porous rock, whereas water pooling in soft clay basins was prone to stagnation and bacterial blooms. If a clean spring was unavailable, your ancestors manufactured their own geological filters. They dug shallow holes in the sand 3 feet away from a murky riverbank, allowing the river water to percolate through the natural sand bed, stripping away sediment, large parasites, and organic debris before scooping up the clear, filtered water from the pit. What about microscopic parasites like dysentery, cholera, or giardia that leave no obvious smell or color? How did ancient humans protect themselves against invisible pathogens before germ theory was discovered in the 19th century? This brings us to a fascinating, counterintuitive physiological adaptation: the human tongue and the evolution of taste perception. In evolutionary biology, taste is not an aesthetic luxury, it is a life-saving chemical analysis engine. Your tongue is packed with specialized taste receptors designed to detect chemical dangers before they enter your stomach. Sour and extremely bitter tastes trigger immediate disgust reflexes because bitter compounds in nature are frequently linked to toxic plant alkaloids, bacterial waste products, or heavy mineral contamination. Research led by evolutionary biologist Dr. Paul Breslin at Rutgers University demonstrated that human bitter taste receptors evolved specifically to identify harmful toxins and spoiled organic matter at extremely low concentrations. When an ancient human evaluated an unfamiliar water source, they conducted a microscopic taste test. They did not gulp down a handful of water. They placed a single droplet on the tip of their tongue. If the water tasted flat, metallic, bitter, or sharply sour, their brain instantly triggered an involuntary spit reflex. This chemical detection system was further reinforced by natural cultural habits. Early humans learned to boil water using hot stones long before the invention of metal pots. Archaeologists excavating Upper Paleolithic cave sites in Europe, such as the famous Chauvet Cave in France, have uncovered fire pit hearths containing heat-cracked river stones alongside water-holding leather pouches and wooden troughs. By dropping red-hot stones into leather skins filled with water, your ancestors brought the liquid to a rolling boil, unintentionally sterilizing invisible pathogens tens of thousands of years before Louis Pasteur proved the existence of bacteria. Now let us examine a mind-blowing, counterintuitive scientific twist that completely flips our modern understanding of water security. We modern humans look back at prehistoric hunter-gatherers with pity, assuming they lived in constant misery, drinking filthy water from mud puddles while we enjoy total water safety. But historical and anthropological data reveal a shocking truth. Nomadic hunter-gatherers actually suffered far less from waterborne epidemic diseases than settled modern civilizations. To understand why, we look to the landmark research of anthropologist Dr. James Sussman, an expert on hunter-gatherer societies and author of extensive studies on the Khoisan people of southern Africa. Dr. Sussman demonstrated that nomadic hunter-gatherers maintained exceptional water hygiene precisely because they were mobile. Small bands of 20 or 30 people moved continuously across vast territories, never remaining in one campsite long enough to contaminate their local water supply with human waste. They naturally protected their drinking sources, establishing strict cultural taboos against urinating or defecating near upstream springs. The real water catastrophe in human history did not happen in the deep prehistory of hunter-gatherers. It happened when humans invented agriculture and settled into permanent towns and cities 10,000 years ago during the Neolithic Revolution, when thousands of people built permanent mud brick homes beside standing rivers and irrigation canals. Human and animal waste inevitably seeped into the local water table. Irrigation ditches became stagnant breeding grounds for parasites, cholera, typhoid fever, and dysentery. For the first time in human history, humans were forced to drink their own recycled pollution. The invention of agriculture trapped humanity inside a permanent water crisis that lasted for thousands of years, leading to devastating urban plagues that wiped out entire cities. Ancient hunter-gatherers, with their forensic tracking skills, possessed far cleaner, safer water habits than the kings and citizens of ancient Rome, medieval London, or 19th century New York. When you mirror this ancient evolutionary history onto your modern life today, the contrast is astonishing. Today, we live in a world where water is completely abstracted from nature. We push a lever, buy a plastic bottle from a vending machine, or pour a glass from a filtered dispenser without having any idea where that water originated, what rock layers it filtered through, or what ecological systems supported it. We have traded our deep sensory connection and forensic observational skills for invisible municipal engineering. Most modern humans cannot read a single animal track, cannot identify a single water filtering plant, and would walk right past a deadly toxic pool without noticing a single warning sign. Yet deep inside your biological architecture, those ancient forensic instincts remain dormant. When you smell a faint, musty odor in a glass of water and instantly feel a wave of hesitation, that is not an irrational annoyance. That is your ancient evolutionary survival engine kicking into gear. That is the same chemical sensory system that protected your ancestors on the African savanna 50,000 years ago, warning you that something in the environment is off. Your body still remembers the vital lesson that kept your lineage alive through hundreds of thousands of ice age winters and scorching dry seasons. Water is not just a liquid, it is the ultimate record of ecological health. You step back into your bright, sunlit modern kitchen on a quiet afternoon. You hold your glass beneath the chrome tap once again, watching the clear stream of water fill the glass to the brim. You lift the glass to your lips, but before you take a sip, you pause for a brief moment. You look at the crystal clear liquid, appreciating the incredible journey it took to reach your hand. You realize that your ability to drink clean water today without fear of death is not just a triumph of modern engineering. It is the continuation of an epic human story that began with broad-chested, barefoot trackers standing on dusty savanna ridges, reading animal prints in the mud, testing single droplets on the tips of their tongues, and learning to decode the secret language of the earth. You take a cold, refreshing drink, feeling a deep, quiet gratitude for the ancient forensic wisdom that kept your ancestors alive, preserved your lineage through the darkest valleys of prehistory, and carried the human story forward to this very moment.